Glad to have you all here. We're really happy. My name is Mike Allen. I'm director here at the biological station, and I'm very glad to have Dr. Luke Griffin here. Luke and I have we have a lot of shared colleagues, but haven't gotten a chance to spend a lot of time together. We got out on the boat yesterday on the Swanee River, and it's been great to catch up with him. I think there's a lot of potential connections between the work that Luke does and what we do here. I've been wanting to do tarpon work here for a while. We haven't been able to do it out at the biological station, but a lot of other projects as well. So Luke he did his undergraduate at the College of Charleston, and he did his master's and his PhD at University of Massachusetts at Amherst. And he's worked literally all over the world on recreational fisheries issues. And today he's going to talk to us about tarpon. And I know he's spent some time looking at his data as well with regard to Cedar Key in particular. So Luke, thank you. Very glad to have you here.
Thanks for having me.
You're welcome.
Those folks online can hear me talking.
I think so.
All right. Thank you so much, Mike. Yeah, it's been a real pleasure. I've been hoping to come up here for some time. I'm relatively new at USF in Tampa. I started there August 2024, so still getting sort of on top of the landscape. So Mike posted me, and I am excited to try to collaborate with you all and make a couple more trips up in the future. Now, so for those that haven't gotten this QR code, I'll post it up again, but it's a lot of these detections from tarpon that were detected in Cedar Key. So don't feel like you're going to miss it if you haven't scanned it yet. All right. So, yes, let's take a poll here. Who has actually caught or fished for tarpon?
What does caught mean?
What's not, right? Baby or big one.
Hooked them.
Hooked them, okay. Good. So, for those that maybe have not, these are amazing fish. This fish, you can see the little fly right here. They love to jump when you hook them. They're absolutely amazing trophy fish. Mostly catch and release. There is a little bit of harvest, but in the U.S. we'll consider these a catch and release fishery. Now, what I find so fascinating, of course, I do love to fish for these, but you can go and target these off of, Florida Keys and this nutrient-poor water where you're actually sight fishing for the tarpon, or you can go up to, say, South Carolina and fish them in spartina grass in the back, and you have these nutrient-rich waters where there are all these foraging migrations.
And where was that picture in the top right?
Top right, that's probably the Florida Keys, I want to say. The mangroves and the flat systems. Yeah.
Did they get real big?
Oh yes. Yeah. We'll get into that. Yeah. Imagine 6 feet, no problem. Yeah.
They don't hurt people.
Well, no. Unless when you try to grab them, sometimes they'll jump up and hit people. But otherwise, if you don't mess with them, they're not going to mess with you. Yes. And so they have all these different ecosystems that they're across. This is their range. You can see it's really vast in the Western Atlantic. They also occur off the Africa coast there. But this makes it really challenging for conservation and management of tarpon when they have such a large range. And when we think about tarpon, we think about the Southeast U.S. really being the hub of the tarpon fishery. In Florida, this is not just culturally important, but economically, right, worth multiple hundreds of millions of dollars. But back in the day, really, it was Port Aransas in Texas. This was the tarpon capital of the world. You can actually go to this tavern, see all these scales on the wall that people have caught this tarpon. This is FDR's in 1937. This is one of my favorite papers where they reconstructed the fishery based on these scales, the age, because it's just like a tree ring where you can sort of guesstimate about how old a fish was and what size. Now, in the 40s, 50s, and then the 60s, this fishery collapsed. There's a lot of not necessarily a perfect understanding of why it collapsed, but certainly one of the big issues is we were catching these fish. We weren't eating them, but we were just throwing them over at the dock. Now, these fish take about 12 years to mature. And also, they live up to 70 years old. So these are qualities of fish that once you remove one of these, it takes a long time to replace that individual. So it's a bit of a cautionary tale on Florida. If we lose this, what kind of devastation would that be for local communities or coastal communities? So working with Mike and a past UF student, we've actually started to use some of the simulations and said, well, how long does it take for a tarpon population to recover at such a X number of percent depletion? But at 80% depletion, it takes around 40 or 70 years. And actually, the Texas fishery is starting to come back online right now. So it's a very long time for this fishery to recover. Again, that's why we want to get ahead of this potential issue if there's any declines. A few years ago, we released a survey to anglers and guides, and we asked their input about a range of topics about conservation management priorities. One of the questions was, well, how has the adult tarpon fishing quality changed since you started guiding and fishing? Because there are no stock assessments. We don't understand the population. We have to rely on anglers and guides entirely when we're dealing with these catch and release fisheries. So we got this feedback. The majority said greatly declined and declined. But in the blue, you see improved and greatly improved. That's a bit surprising, but that's nice. We dove down a little bit deeper in the demographics of this, and we could see those that had been in the fishery for upwards of 2, 3, 4 decades. That's really this category here, whereas the newer generational anglers might have seen an improvement in the last 5 years. And at least in fishery science, we call this the shifting baseline syndrome, right? Daniel Pauly effect, where we only know what we experience, right? So what are the actual threats for tarpon? We have coastal development, we have pollution, harmful algal blooms, destruction of nursery habitats, the interactions we have with the fish, not just if we release this fish, does it survive or not? That's a big question. And then more pressing, there's more concern about the overharvest and potential exploitation of their forage fish in the more northern areas on their migrations. So I'll break down these threats. But to really make sure we're focusing on the how we're going to better protect these fish, we have to understand their life cycles and how do we know this information about each of these phases. So tarpon, they go offshore to spawn, primarily in the early spring, but all the way through August. They dive down to 300 feet and release about 20 million eggs. And they have this unique larvae phase called leptocephalus. It's shared among American eels, bonefish, ladyfish, and tarpon. For 1 month, they're floating in the open ocean. They come back into these nearshore areas, and then we get to see what a little baby tarpon looks like, about the size of your hand, a little smaller. And they're in this phase for a couple years until they make their way a little bit further out, and they take around 12 years. That's very highly variable to mature. And that's between around 50 inches when they reach maturity. So to answer your question, they can get up over 280 pounds, right? 3 to 8 feet long, and they can live over 70 years old. And these are very highly migrant, going up and down. So how do we know about where they spawn? Well, we have a lot of question marks out there where we're still not sure where tarpon are spawning, but we also have these blue bubbles. This is where we're suspected of spawning. Now, we can do this in a couple of different ways. We can backtrace where we find the leptocephalus larvae and current patterns, but we can also look at satellite telemetry data. Now, this is a great article by Liu et al. They had about 200 fish tagged with satellite transmitters tracking these fish. And from depth 0 to 360 feet on the y-axis, this is just for one fish. This fish was tagged around May 12th. It's an old one, 2004. But you can see this fish is using around 0 to 75 feet in depth. And then as we approach the new moon in May 19th, it dives down to 300 feet, comes back up. And this is very classic of a tarpon spawning, right? So even though they're a nearshore fish, they spend over 80% of their time in less than 10 meters. This is a very characteristic of spawning. That's kind of how we can help piece together where they're spawning. So leptocephalus larvae, they'll actively make their way back to near shore. They use different parts of the water column for transport, but also a lot of passive drift mechanisms through the loop current to make their way back. Now, some of the threats that the larvae will face are these oil spills. You can think of the deep water oil spill in 2010. Now, that could have easily knocked out multiple age classes of these very fragile larvae, right? We're just not sure exactly what the damage did, but 12 years of maturity, we might be starting to see that effect now as well. And so there's not just the Deepwater Horizon spill, there's others. Even this one, the Taylor oil spill, is still ongoing. There's a lot of unknowns about what's happening out in the open ocean for these larvae. But for those that are lucky enough to make it back into near shore, well, they might encounter these harmful algal blooms. I'll get more into this details about Carina brevis, but again, highly fragile larvae. These blooms occur right when the tarpon are coming back in. We had a really bad red tide in 2018, especially in Tampa and Charlotte Harbor. But we also have blue-green algal blooms as well. Okay, so we have our baby tarpon, it's transforming, and then we get to the perfect nursery habitats. We like to say if you can find a really difficult access point and get all the way back here, and it seems impossible that there's going to be a fish back there, that's really nice tarpon habitat. They're going to hang out back here for a year or two. And what's interesting is this is very low oxygen. Now, for those that do fish for tarpon, it's really neat to see them roll, right? They go up on the surface, they come up, they take a big gulp, and then they go back down under. Now, they do that because in their juvenile phase, they're in these low oxygen areas where they're able to hang out here, gulp atmospheric air. This helps them reduce the competition load across other species and also avoids predators that might be consuming them. So this is a very neat evolutionary trait about tarpon that I just love from their juvenile phase. So frameworks of solutions for juvenile tarpon. Now, it might be kind of hard to see here in the light, but this is prime tarpon habitat for juvenile fish. It's right next to golf courses, right next to development. People see critical and important tarpon habitat and they say, well, that'd be a great place for a condominium, right? So what can we actually think about? Well, freshwater flow management, we have issues with Caloosahatchee, reducing nutrient runoff that fuels algal blooms, wastewater infrastructure, septic to sewer. I was amazed to hear that this is all sewer in Cedar Key. That's really great. Florida Keys is a little bit behind on that. Treatment plant upgrades. We've seen pharmaceuticals affecting redfish, bonefish. We haven't tested in tarpon, but I'm sure it could certainly be part of their what would you say? Issues, I guess. Protecting natural habitats, incorporating that into the actual development plans, and then habitat restoration. And I've been enjoying some conversations here, not just doing habitat restoration, but how does that actually influence the population numbers after you enact some of these restoration operations. So I kind of went a little faster on the juvenile phase. I'm sorry, I'm a little biased. I love the adults. So I'm going to talk about some of the same threats in different contexts that affects the adults. And this is really where I started with telemetry and tracking their movements back in 2015. And we started tagging these tarpon small 10-pounders all the way up to 150-pounders. And you put a small, AA size battery in one of these fish, and it swims by one of these receivers, which there's around 30 out here in Cedar Key. You get a detection. And then Mike and you all would share that detection. Those folks in Louisiana would share their detections, and we can backtrace their movements up and down the coast. And we work a lot with different fishing guides as well. So I'm going to summarize a lot of what we just talked about in the animation. Instead of going too much detail into the graphs, but what I want you to understand is there's these two different migratory contingents, one that migrates up the west side of Florida, one that we've seen migrate up the east side. There is some crossover, but large and large, they're really repeatable migrations. If you stick to one side, you're going to go to that side, and you're going to the same location as well. So we have these two different groups mixing the keys. These are going off to shore to spawn. These are going offshore to spawn. They release their gametes. They get those leptocephalus larvae. They make their way back to the coast. This is why we don't have too much genetic variation, we're thinking. They come into these nearshore areas. Again, they're up here in these fragile habitats upwards of 12 years. And then they make their way out towards those nearshore coastal areas, and they get intercepted by the migrating adults. And we think it's a social facilitation. They're teaching those younger juveniles where to migrate. And then what we see in the telemetry data, they will migrate to the same location year after year, almost within the same week period. It's fascinating how highly repeatable these migrations are. Now, they don't all go to these northern areas as far. It's just demonstrative purposes. But they are going to these northern areas, including Cedar Key, for these foraging migrations upwards of 4 months of the year. And then you can see the Western Gulf is actually mixing here, and there is some spawning off here. Now that's under natural habitat conditions. As we know in Florida, it's ripe with issues, right? We have issues with freshwater flow, sugar industry. We have issues with sharks. It's a very much growing concern. We have issues with coastal development, destruction of watersheds. And as these fish are migrating, pollution that fuels harmful algal blooms. And then when they get in the northern areas where there's nutrient-rich waters, there's a concern of overexploitation of their forage fish, primarily menhaden, and this is a growing concern. So even though they're considered catch and release fish across their juveniles all the way to their adult phase, it's a bit of a death by 1,000 cuts, which might be leading to somewhat of a reduction of the fishery compared to, say, 50, 60, 70 years ago. So how can we use this data and actually enact some management practices? Well, usually, even though Florida, we have this great fishery, the fish that's caught up here is going to be traveling up and down the coast. And we manage tarpon at the state level, unlike other fish. And so we have different regulations, say in Louisiana, there's a tarpon rodeo. There is a harvest, but again, it's not for consumptions, but it's for tournaments. It's relatively minimal. But we have other states that have different regulations depending on how many fish you can keep, what size of the fish you can keep. We have our medium level fish policies, and then we have our catch and release states, that being Florida and very proactively Virginia. And so our fish is that same fish in Louisiana. And so it should have a cohesive management framework, right? It only makes sense. And so with this data, we are able to push forward a grassroots initiative and a catch and release amendment in North Carolina, which we hope that would take over the rest of the states in recognizing the importance of this game fish. Now, Florida does get a gold star because they are the only state that has a catch and release handling guideline built into the policy. The fish is above 38 inches, it has to remain in the water. Now that's really important because we've been seeing that these fish are traveling multiple hundreds of thousands of kilometers, right? Or multiple thousand kilometers. And it's very energetically expensive for these fish as they migrate, right? So you get caught a couple of times, there is post-release mortality. People think of them as this really robust fish, but they are fragile and they do die after you catch them, in especially with warm waters. So what does that actually do to the reproductive output? Is a big question. So I'm going to jump into the Florida Keys. This is where I spent most of my time. And this is again with the mixing areas between the west side of Florida and the east side of Florida meet before they go offshore to spawn. Those white dots are the acoustic receivers that we're, with collaborators, we're downloading to get those detections from the tarpon. And those green arrows and those green lines, you can think of those as sort of main pathways between these receivers. You can think of them as highways almost. The thicker the arrow, thicker the line, those are more connections between those locations. So certainly along the ocean side of the Florida Keys, it's hard to see, but here's all the keys in the background. And US 1 goes here. Now, we talked about the mixing areas, really important, and repeatability. We see that where the same fish is going to show up to Cedar Key, pretty similar time every year. It's remarkable if you see a fish and it arrives the first week of April, the following year, it's going to be the first week of April. If you have a fish that arrives, say, the second week of May, it's most likely that fish is going to arrive the second week of May. It's not just temperature related. Same with overwintering, same fish. So we think what's happening is maybe there's another environmental cue out there, like photo period, like birds, that cue when to migrate. Certainly around 74, 75, 76 degrees is when you get the mass of the fish, but they are showing up in a very repeatable time frame before they move offshore to spawn and they're in these channels. So we did this paper a few years ago. Mike was a part of it and looking at climate change and the cascading effects all on tarpon. I'm going to start to pick apart this graph and look at some of the threats just to isolate them and how our data addresses. So the first one I'm going to look at is the interactions we have as anglers with tarpon. And we have been getting an increasing and larger issue with predator-prey interactions. Now, in Boca Grande, you might hear of bull sharks, or hammerheads. In the Keys, most of it's primarily hammerheads. There are some bull sharks, but hammerheads is a big one. Now, this fish is actually, you can't see it, it's actually connected to a fishing line. So that shark is removing that fish before they even land it, and we call that depredation. It's happening with sandbar sharks, with other fish, but hammerheads are depredating tarpon quite a bit. And so we wanted to ask the question, well, can we use the data across the Florida Keys to look at how tarpon and sharks are interacting? So you had those white dots before for those acoustic receivers, but we clumped them into these different nodes of these different areas. And the yellow here is the tarpon detections. So larger yellow circle is going to be more tarpon detection. Blue would be hammerheads, and then the overlap with them is gray, grayish color. Same here, but with bull sharks, right? We have red, and then the overlap would be orange. But we didn't want to just look at overlap. We wanted to look at the arrival timing of these sharks when there were tarpon. We want to answer the question, are these sharks non-randomly moving around to these different locations and potentially looking for opportunistic foraging, right? And so it was pretty clearly obvious that they were. It was not random, right? And particularly, it was in areas that was tarpon angling locations. And these big pre-spawning aggregations when people are fishing them intensively and the sharks come in when the tarpon were there. And so we're going to dive down to that one arrow. This is across the Keys, but we really focus on Bahia Honda. Has anyone been to Bahia Honda? Yeah. Has anyone caught tarpon there? Yeah. Okay, Mike. Yeah, nice. So here's our receivers. I'll reference you all, and we're going to zoom into here. Here's US 1. Here's the old railroad bridge. We actually had receivers in between because we tagged hammerheads and tarpon. We want to look at fine scale dynamics. And this is a typical setup on US 1 when you have these boats and these bridge spans, and they're drifting crabs back around 10, 12 feet down, looking to hook into one of these larger tarpon before they move offshore spawn. And this is a guide friend. You'll see a hammerhead right here, and they're depredating that tarpon. This is a common phenomenon. So Dr. Grace Castleberry, she was at Andy Dalenchuk's lab where I did my graduate school and postdoc at UMass Amherst. For summer, she sat on this old bridge, this old railroad with a pair of binoculars, and she watched how many times this happened just to get an estimate on how bad post-release mortality, depredation was. We can't get post-release mortality because that's typically under the water, right? And so she found that if you caught a fish and you were fighting it for more than 5 minutes, there was an estimated rate of 15% mortality minimum, right? That's what she saw. So whether that's going to actually be a population level impact, we still don't know, because we don't know how big the tarpon population is. But again, these fish can be upwards of 50, 60 years old before they move offshore to spawn. So certainly talking through this is, what do you do? Well, some guides are moving to a no-touch policy where you can break the line and let the fish go. Or they're using heavier tackle to bring in the fish much quicker. Or if there's sharks, maybe modify where you fish. But culturally, this is so important, and economically, you have to find a solution working with fishing guides as well. And we found generally when the tide was going out, that was really when the depredations were the worst. And we have movement data that shows that hammerheads are positioned right underneath the tarpon, depending on the tides. So we had those receivers out. We tagged tarpon, we tagged the hammerheads. Here's the acoustic tag. We did it externally for the hammerhead. And we put these cattle ear tags on for identification. Right, so we had these different color combinations, and we've worked with the fishing guides, and they'd tell us, I saw white, blue, purple yesterday or whatnot. It was amazing how few we got. We had a good relationship with these fishing guides, but we had very few of those guys saying, yeah, we saw these cattle ear tags. Well, we saw another hammerhead. They didn't have any cattle ear tags, which was telling us there was a lot of hammerheads that were coming in and out of this area. And interestingly, the vast majority of these were females, really round females. We actually did ultrasound. You can see the pups, and so we're starting to think that's a reproductive opportunity, right? For reproduction, getting those extra calories from tarpon. You dive down, and you'll be able to find these tarpon scales at the bottom of the ocean. So we looked at the telemetry data on the y-axis here. We had the mean residence time of, how often or how many minutes were the tarpon staying inside those bridge area. From January to December, it's relatively even because there are some subadults and overwintering, so it's relatively flat. But then when you actually look at the hammerheads, March, April, May, June, that goes straight up, right when the tarpon arrive. Let's look at the hour of day. So again, mean residence time in minutes in between those bridges. We have night, day, night, corresponding to the hours. Tarpon is relatively consistent. They're always there in that channel. But for hammerheads, it goes up during the day and then goes down at night, which coincides perfectly with the time of fishing, right? So these are habituated fish, right? And sharks are fish, so it's habituated sharks there. So back to our threat map. I'm going to zoom into this little dinoflagellate here. It's supposed to represent harmful algal blooms. We had a really bad one up in St. Joe Bay last October, but in 2018, when we were looking at this sort of really bad one, Tampa to Charlotte, this is Carina brevis, and they have these brev toxins that can be neurotoxins, right? Not just to tarpon, but to other game fish like snook, whale sharks. It affects our human health, marine mammals, and it's naturally occurring. But as it gets into near shore areas, you can get these fuel nutrients that really help and increase the intensity. So we wanted to understand, well, how is it interacting with tarpon? So we had the fish kill hotline from FWC, and here each red dot is actual mortality reported from harmful algal blooms. And the brighter the colors are, is the higher fish kill count from different counties. We looked at telemetry data and understanding, well, are tarpon moving in and out with the harmful algal bloom, or are they not affected? And lastly, we looked at tournament data to look at if you were to catch more fish during or without elevated algal bloom. But most importantly, we also talked to the fishing guys at these semi-structured interviews on what's their perceptions of how HABs affect tarpon. And so they really conclusively said blooms are increasing during peak tarpon season. And interestingly, they said that they would see tarpon foraging along these harmful algal blooms on the bait fish that are spinning up. And it didn't seem like it affected them. And compared to other species, there was a lot less fish kills than it was for tarpon. And so, yeah, fish kills do occur, but what's happening reproductively if they're feeding? We know Carina brevis affects the reproduction output for other species. And for telemetry, we didn't see any difference of movements with the HABs, but the guide said if it gets intense enough, they will leave the bed. The fishery impacts from the tournament data, it made sense during an elevated algal bloom. You catch fewer tarpon than when it's not elevated. And so this is going to really impact this $100 million fishing industry. And all this is to say there's evidence of reduced nutrient discharge. Okay. So forage fish, this is a growing concern in the Northern Gulf. Menhaden. At least at the Atlantic side, there's ecological reference points actually spearheaded by you all now, but traditionally menhaden has reduced quite a bit, upwards of almost 90%. That's the Atlantic side. The Gulf side is another issue. Last year, there's some footage of bycatch of tarpon in Louisiana. There was a big effort to make sure there was buffers for the menhaden fishery, which did not happen. So there's a lot of debate between not just anglers and management, but also across scientists on how important are menhaden for tarpon. So we did this study where we asked fishing guides to collect these fin clips all across their range. And we did this stable isotope analysis that really helps us understand how connected are these ecosystems. What are the primary foraging channels for these tarpon? And I'll explain exactly what isotope analysis is. But we have a large swath of where we got these samples. So how it works, the simple analogy we always say is you are what you eat. And so that gets incorporated into your actual tissues. And if you take a sample from your hair or a fish's fin clip, we can understand a bit of your diet, right? That's how we understand humans from, say, 1,000 years ago were eating meat, vegetables, or fish. We can do the same concept with tarpon. And if you do this across a large enough space, you start to understand a bit more about their diet preferences and energy channels. Now, the issue with tarpon is you take that fin clip, you're actually representing 3 to 5 months prior where that fish was foraging. So even if you sampled a fish down in Southwest Florida, they were feeding off Menhaden, you have to figure out where they were feeding based on where you think they were 3 to 5 months prior. And that's where the telemetry data really helps out, and we can build these mathematical models to try to guesstimate on where they were getting the resources in the prior months. So I'm just going to walk you through a couple examples of what we found. So this was a 70-inch fish captured in October off of Louisiana. You can actually see this red dot is where we caught it. We built this based on the fin clip information and also the movement data. And it's giving us a probability map of, you can call it the most likely area that fish was foraging in. The brighter the color, that was more likely where that fish was foraging in the months past versus the darker color. It became pretty clear that for this fish, the Northern Gulf was a really important area that that fish was foraging within months prior, which makes good sense. Now, if we have a 55-inch fish captured in April down here in the Keys, we can do the same probability surface, but we don't know if that fish had migrated west of Florida or east of Florida. So we have to allow the likelihood surface to go up and down the coast. But what I want you to understand is you see the bright colors. Again, it's these northern areas that are probably part of its foraging migration in months past. And if you do this for every fin clip that we actually collected, and you say, draw an arrow to the most likely place that fish was getting its resources. Now, I know that it wasn't exactly at this arrow, but it is certainly pointing you to across these different ecosystems that these fish were depending on. So this is really evidence for us to say, hey, Louisiana, for your Menhaden fishery, it's important for us down in the Florida Keys that are catching those tarpon. Now we can look at also what kind of forage fish they were feeding or invertebrates, and this dominates this Clupidae forage fish, such as Menhaden for these tarpon. But we don't have the exact species-specific information. It's, in this analysis type, it's very sort of broad strokes what we said. And so right now, what I hope to do with folks that are online or those in the room that catch tarpon is we're going to get these Q-tips and we're going to actually insert them into their rears, get the feces from them, and we can use DNA to pinpoint exactly what prey items those fish were feeding on. So we don't have funding for it, but I do have some kits to get some of that data for some keen anglers to put this Q-tip inside a tarpon. And this is sort of that missing link that we need to bring to the Louisiana State Agency and say, Menhaden is, or maybe it's not important to tarpon with their foraging. So I mentioned that warming sea surface temperatures, this is interacting with all these different threats. I'm going to dive into, how might climate change affect the fishery for tarpon? Now, the first question we have to ask is, well, what are the preferred temperatures? What are the optimal temperatures? What's too hot? Again, we're going to use this Liu et al. That tagged almost 200 fish with satellite telemetry tags. And from those satellite tags, you can get exact temperature readings. So we have temperature in Fahrenheit all the way up to 91. Each black dot here is a temperature reading across the months. And the red boxes is just to show you how those points are clumped, right? Those are histograms. And so we're using this graph, we're just going to look at it and eyeball it and say between 68 and 84 degrees, we consider this the optimal temperature range for tarpon. This is what they prefer. Tolerated would be 84 to 90 degrees. And then above 90 degrees, we call that too warm or too cold, under 68 degrees Fahrenheit. So with our classification, we collaborated with climatologists from France. We're in the process of doing this, but we wanted to ask the questions. I'm going to walk you through this slowly, and maybe I'm going to walk it through twice because it's confusing when you first look at it. We wanted to ask the questions, how many days in a given year in a certain location does it fall between those two temperatures, between 68 and 84 degrees Fahrenheit? So we're focusing on just this one region, just for this plot. We have it across the regions, but we're focusing on South Florida. We're taking the temperature readings from this area specifically. And on the y-axis is number of days per year that fall between 68 and 84 degrees Fahrenheit. And on the x-axis, it goes from 1950 to 2100. I want you all to look at this little black line. And this is tracking how many days between 68 and 84 degrees Fahrenheit from 1984 all the way to present day. This is actual real data that we were able to collect from satellites from this blue little shape. And you can see it's decreasing number of days across time for the last, say, 40 years. But we didn't want to just stop there. We wanted to understand, well, what did it look in the past? What was it going to look like in the future? And so we took climate models that are developed by much smarter scientists than me. And we applied and calibrated the observed temperature to these climate models. And we projected out how many days we're going to observe in this optimal window. And you can see each different colorful line is a different simulation under a different scenario. And you can see as we move down towards 2100, it's about a loss of 10 days per decade of optimal conditions in South Florida, right? And we can see this in real data and then also in the projected data. So what does that mean to our fisheries and how we actually manage them is the next question. So we can look at this at a little bit of a different graph. I like this, mostly because of colors, but we say number of days per year again, 350 to 365, excuse me, to 0. And then year, this little white line is us, 2025 out to 2100. And you can see, again, the optimal conditions for one of these. This is just pulling out data from one of these colorful lines, one of these models. And you can see the blue line, optimal days start to decrease through 2100. The tolerated, there is a large band here, but it starts to decrease in about 2060. And then above 90 degrees Fahrenheit, you see it starts to pop up and how frequent it is around 2050 all the way out to 2100, where it gets really warm and predicted at 150 days at 90 degrees Fahrenheit. Now, is this going to happen? That's another question, right? We're slowing the emissions right now, or at least that's the hope. This is the worst case scenario. And if this is going to happen, we're going to have a lot bigger issues than just catching tarpon in 2100. So how do we actually prepare for this? We made this argument about habitat management restoration is a good safeguard to help support these fisheries. And this is a missing element quite a bit in the flats ecosystems beyond seagrass. And we wanted to provide a sort of guiding principle list, 10 guiding principles focused on flats fisheries and flats. And I'm not going to go through each one of these, but the two biggest ones I'd like us to all take away from here is we should consider tarpon as an umbrella species. If you protect tarpon, you're protecting way more than just one game fish. You're protecting all the wading birds, you're protecting the snook, the spartina, the mangroves. If you apply this to all the life cycles across tarpon, it's a really, really nice umbrella for the other species, right? But importantly, that's great, but you have to make it sure it's a flagship species, something that we can all behind, right? We see these NGOs, non-government organizations, guiding associations, angler associations. You see mailboxes in the shape of tarpon. People know what tarpon here in Florida, so we can get all behind conservation and management of tarpon. I'm going to wrap up the talk here. I'd like to first acknowledge all the amazing partners that have helped out this work, especially off of here in Cedar Key. I'll have that QR code for folks if they're curious about seeing the detections of tarpon off of Cedar Key. But really what I like to highlight is working with the fishing guides and angling. These are just a handful of folks that have helped us in the years past. Hunter, when you were there, maybe 2017, much younger.
Yeah.
With Brian Sawyer as well, helping us tag fish. Not only do they help us collect the samples and actually catch these fish, but they're the ones that are driving the questions that we're looking to answer and provide empirical data that state agencies often need to actually enact policy. So really, really important as well. So with that, I'll say thank you and thanks for hosting me, everyone. Yeah, appreciate it.
Awesome. All right, we've got time for questions. I'm sure we're going to have some, and let me grab the mouse and I'll be able to look online, but we'll open it up in here first.
Maybe just press on that accordion too.
This one.
Yeah.
Jerome, nice.
Oh, sorry. It was just kudos. It was just congratulations for the presentation. It was fantastic, and thanks for presenting the results on our project on climate change.
Yeah, thanks for showing up. They can ask you those questions now. Yeah.
I was struck by the photoperiod sensitivity of tarp migration when you're talking about all these other temperature effects. And so just curious whether I think it's photoperiod or temperature that will drive long-term patterns of behavior, and also how like maybe the adults are moving based on photoperiod, but maybe their larvae rely on temperature or something else, and kind of how you think that.
Yeah, and the Florida Keys would be a mismatch of temperature and timing of the migrations is a concern of being too hot. But at the population level, I do think temperature is the main driver. Most of them will arrive and migrate with the 76-degree thermocline or change in the temperatures. But again, at the individual level, I can't think of any other variables that would drive it because it's showing so reliably every year. But yeah, I think that's a big concern. How does it match with the leptocephalids that are being spawned offshore? How does it relate to the pre-spawning aggregations? At least for the fishery, if they're going to be still spawning at the same time, say April, May, and June in the Florida Keys, and they're going to stay there even though it's getting warmer. Well, if we catch those fish, they are super fragile, around 87 degrees Fahrenheit. That's when they sort of go up to the side and sink off, and you don't know if they survive. So the temperature stuff is also related to catch and release handling practices. I kind of took that on a different angle, but yeah.
Other questions?
Hey, Mike, I have a question.
Shark deprivation here, we always hear about Boca Grande. We hear about it in the Keys. Do you see it up here as well, or do you not have much data? And that might be a question for Mike.
Yeah, I would ask the room for you all. Yeah, I haven't
We've
yet.
definitely seen it. Yeah, I mean, the places that we catch tarpon with regularity also have a lot of big sharks. So I don't. It's not like I've seen a lot of hammerheads eat sharks, but you know what happens as soon as you release that fish, you don't know. But I can tell you that they're being caught in areas where there are a lot of big sharks present.
And I've noticed with bull sharks, post-release mortality is a bigger issue where you can see them on the sonar. They're just there waiting until you release the fish. So,
Because we know we've trained our shark population very well for red snapper, especially here in our area.
Yeah.
And I wouldn't be at all surprised to see that we've trained them up on tarpon.
and
I don't know that we target as a fishing community tarpon as much here as in other areas, but I'm sure it's still happening. I mean, Hunter, you're a guy. Do you?
Yeah,
see that?
Yeah. I'd say I've never personally seen it here, but I've seen it everywhere else from the Keys to Tampa Bay, and Homosassa has it a lot with like tiger sharks and bull sharks.
Okay.
I just don't think there's not enough people fishing for tarpon around here
Yeah.
for the sharks to learn it. But I definitely agree that there's a possibility because where we're fishing for tarpon, you're seeing a lot of big sharks as well.
Yeah.
I think it's definitely got a lot of potential in the future to start happening. They just don't know it yet.
maybe a one-off here and there, but yeah, like you're saying, Tampa Bay and big numbers, the sharks are just waiting. In Texas, it's less of an issue where they're fishing for tarpon in Texas. It's, a few miles offshore, and it's much more spread out, and so again, it doesn't condense all the fish. But I wouldn't be surprised if it becomes an issue everyone.
Yeah. Awesome talking to him. Really, really cool stuff. The animations were incredible. You put the rest of us to shame. Really, really good. I'm curious about. I mean, they're such a unique species. I'm curious about when they're young and they're up creek, right? They've got a home range that, or at least they're spread throughout the whole marsh mangrove tropicalization gradient. And I'm curious. You know a lot about this species and what they're doing at different points in time. Do we know much about what their diet's like when they're way upstream like that? And are there differences in marsh dominated, mangrove dominated? And then is that changing over time? So 3-parter.
Yeah. I don't know what their diet is. We always say inverts and small fish.
Okay, yeah.
And it's definitely going to change depending on what system you're in, because the range of maturity is going to be probably influenced by that juvenile life phase on where they are. So it's a great question that we're not sure about. And also just, are these fish surviving at some point, right? 12 degrees Celsius that's when they're going to croak. And so we see juveniles up in South Carolina now, and some of them are surviving these deeper holes. But at some point, we assume they're going to start contributing to the population. We just aren't sure. But I'd love someone else to take that on and explore it here.
Got a couple online, but go ahead.
Yeah, this might be more of a question for Mike, but in the research that the grad students, you folks have been doing on snook up in the tidal creeks, did you find many juvenile tarpon with there?
I'll ask Robbie. Robbie's doing that work.
That's all.
Not horrible, not as big. Yeah. Not enough to really relate that to environmental variables, I don't think, but over time, if we can collect enough of them, yeah, we can start to see what types of habitats they really like. But
Yeah, suspect you guys are going to be studying a lot of juvenile tarpon in your future.
She said Charlie did caught tarpon back in.
Yeah, there's been a few. Yep.
we actually had one in a touch tank at an open house.
Oh, yeah.
Yeah.
Man, even at College Charleston at the underground, we throw a cast net and you come up and big little tarpon. And go, how is this here? So we'll take a question online. Can I ask a question about what drives the adult tarpon to return to the same northern places every year in summer? Is it imprinted? Do they return to the same place every year, more or less? Or do they simply just move north as water warms and simply stop where there is food? Yeah, great question. I'm not going to know the exact answer, but what we do see is they do go to the same locations year after year in the north. I'm specifically thinking of a couple places in South Carolina where it's like the same fish is going to be on the same pier year after year. So they've certainly imprinted. And our argument was that the juveniles start to turn into subadults, and then just at the edge of maturity, or even right after maturity, they start migrating with the adults, and then that's how that imprinting happens. And we see this with other fisheries. It's called entrainment. And so I suspect at some point, though, they see enough forage fish and they would stop in Louisiana. But Louisiana is also strange because we know that they're spawning off there in August. So it's a foraging migration, but also there's another important life cycle thing they're trying to complete.
It's like Dave's got a
Yeah,
question.
Dave's got one. Yeah, go ahead, Dave.
Hey, Lucas, great talk. I'm curious, maybe it's putting you on the spot, but if you had to rate, the stressors, is which ones are probably most critical for tarpon currently? How would you rank them?
Sure, in terms of like the population status,
Right.
I think there's two big ones you can always come back to. One is the juvenile habitat and just the loss of it across Florida. Those are prime areas for golf courses and changes in water quality. But the other is they're very late-lived fish and long times of recovery. So if we have a larger catch and release mortality than we suspect, that could certainly be driving it. But I always come back to, habitat. Important habitat's the first thing you have to solve. But it's a debate.
Cool. Thank you.
Just an observation. I mean, her observations that you mentioned of 15% of those that she observed, 15% of those had a depredation event. That's a significant mortality source on an animal that lives to 70 years.
That's in one location.
Yeah. So that could be a big determinant of population size.
So we'd like to explore it in Boca Grande, but you have to have buy-in from the community to look into it, because again, they're the ones driving this, and if this is what they'd like to explore, then we're on board. We'll go in the back, and then we'll come back to you.
Has there been any comparisons of the menhaden diminishment with the tarpon? It just seems like I keep hearing more about the menhaden affecting a whole bunch of fish species.
Yeah, I'm glad Dave is actually on this call. You could probably answer some of this, but at least for tarpon, we haven't had that clear connection yet. you hear it from the anglers quite a bit, especially in Texas with that collapse. In Louisiana, you hear about tarpon not being in certain locations because the menhaden have been scooped up in that area. So I don't have any real data yet with it.
Is there any effort afoot to do work on menhaden as far as conservation of it, or is it just?
So Dave, who just spoke, he's doing some great work, and at least for the tarpon aspect, that's why we did this isotope study that points to that, but we really want to say, yes, this proportion is eating menhaden, this proportion is eating blue crabs maybe down around this area versus South Florida.
Okay.
So we're working on getting funding and support for that. Yes.
Is it, you showed very early on the picture of all the scales, and I remember as a kid, that would be the thing. If you were to catch a tarpon, it would be catch and release, but you would harvest one of the scales as your trophy, right? Is that still done, or is that now considered to be bad practice?
I think oftentimes people try to avoid that.
Yeah.
Someone that had never caught a tarpon, I see it happen sometimes. I think it was BTT or FWC, they were doing genetic studies. They were doing the mouth swabs for a while, but also taking a fin clip or scale. But even if you get that scale, you can analyze a bunch of the chemicals inside of it and understand where it was living over its life. But my understanding is it's kind of an older practice that's not encouraged.
Not really done.
Certainly from a scientific point of view, I would say don't do that. Yeah, if that can encourage infection. Yeah.
I guess if you're trying to collect data on the species,
yeah,
but you want to release the fish, that might be a sort of a.
I would say in the name of science, yeah.
Yeah.
Few tips, yeah.
Yeah.
Got a comment about the menhaden? Oh, go ahead.
If the temperature gets that high, so bad for the tarpon, do they actually die?
I've never seen a study on how high they can live in a temperature, but I just know when you catch them above 9 degrees, they're really stressed out and they'll kind of do the slide away motion. But in Texas, they say that's not a big issue, but the water's so dark, you don't really know what's happening after you release them. So there needs to be some more work. But they're fast. They can move quite a bit. So if it's too hot,
They'll just go where it's cooler.
Right.
Had a comment from about Louisiana.
Andrew, I live in central South Louisiana, and our tarpon population have disappeared after menhaden industry ramped up harvest since 1970. So actually, I have a call with Andrew on my drive back to discuss this. I appreciate you showing up here, Andrew.
I'll just add that I've heard from some of the charter captains that have worked out of here for a lot of years that there actually used to be more tarpon here when we had menhaden in the estuary here. And in the last few years, they haven't been seeing them. And I don't know if that's attributable to any menhaden harvest, but it was recognized that there was a link between those prey items being here and tarpon numbers, but we'd still have tarpon, of course. But
my inclination of working with so many anglers is they usually know the answer.
Yeah.
And we just have to find the data to support it.
Yeah. Yeah.
Or do they just switch over to something else? Because
they do.
Work
They
targeting
will.
via recreational cast netting for shrimp in late spring, early summer when they start running. We'll literally see tarpon mashing the shrimp together in a bait pod and start making runs on it.
Absolutely, yeah. They're generalists. They'll eat whatever's there. They're certainly going to the north in those productive waters. But if you go to, say, southeast of the Atlantic side, there might be ribbon fish there because they're abundant. And Biscayne Bay in the winter, they're shrimp, like you said. Worms, polychaete worms, and the keys if they're available, right? And the little below worms. Yeah, blue crab. They're on Homosassa back in the day.
I can't remember from the map. How far north do they go? Do they on the Atlantic coast?
So we've had them go all the way up to Ocean City.
Oh, wow.
But last year I talked to an angler and he caught a tarpon on the south side of Cape Cod, right here, and that's happened a couple times in the last few years.
So maybe because some of our species are coming, we're seeing.
Certainly, yeah, we see more black sea bass in the Gulf of Maine now.
Yeah.
But if you get on that Gulf Stream, they come out of it. They could hit anywhere. But if the temperature's okay, they're going to stay for a bit as well.
Wow.
And we see the acoustic telemetry from tarpon. We'll see early season the Keys. All of a sudden, you get a detection up in Georgia or South Carolina, you didn't have any detections going up. So we think they're going off, maybe spawning once or doing a false spawn to get in the Gulf Stream, to come back in, and they'll migrate all the way back down to the Florida Keys, and then they go back up north after they're spawning in Portugal. So these fish are moving so fast. They're doing these double migrations that we had no idea about. Yeah, Robbie.
If their range is expanding and they're coming all the way back to spawn, have you thought about like the bioenergetics of that?
Just like conversations like this, absolutely, yeah. That's a very far away. And what we see in the winter time, though, instead of doing these very quick migrations as they do in the spring, they'll go like this, almost like a staircase fashion. And we think the fronts are kind of pushing them down. And there's mullet run, at least on the east side. So they are following foraging fish migrations, like mullet down. But yeah, that's a big concern, especially when you add on they're getting caught once, twice, or 3 times. They're fighting for their lives every time.
One more question online. Go ahead, Jerome.
Yeah, Lucas, that's me. Hello, everyone. I probably missed something in your presentation, but do we see areas on the eastern US border where you actually have juveniles, not only like within the Gulf or my underlying question is, could we have spawning grounds and juveniles habitat that will move northward with an increasing temperature, or is it like they are like deeply linked with their both mating areas and juvenile locations?
Yeah, at least on the East Coast, there is definitely juvenile habitat, and those fish will recruit into the adult population, like Indian River Lagoon, all the way up to Georgia. Now, a lot of those larvae and leptocephalus juveniles are coming from the spawn, I think, off the south part of Florida Keys, and those larvae are going up the Gulf Stream and then coming in. I don't know, to answer your question, if they would spawn maybe say off of Cape Canaveral, if that's what your question is. I'm not sure.
Yeah, the main question was I don't know personally, and I don't know if we find juvenile tarpons nurseries in where up north.
Yeah, absolutely. Yeah, so we get juveniles all the way up through South Carolina.
Okay.
I think even North Carolina, they have some juveniles. Yeah, we had fin clips from North Carolina. But one of the questions that Jerome here is the climate scientist from France that was supposed to visit this year with us. But one of the questions is like, can we model the cold snap frequency moving up the coast from past to present and try to map out what is that juvenile habitat and how fast is it expanding with the temperature change? So in the Gulf, we're constrained, right, by the northern part of the Gulf, but at least at the Atlantic, we could test that.
Yeah.
Yeah.
So have you been, do we see them being caught further and further north? I mean, I guess not catching juveniles, but I mean, is that
definitely?
Yeah.
For the adults. Yeah, and sometimes we see a 12-pounder up in South Carolina and go, how did you get there? Right.
Yeah.
They can move.
Yeah.
Sure.
That's cool. I'd be interested in how you guys do that cold snap analysis stuff. Because I mean, it's the same sort of thing we're trying to figure out with mangroves too.
Yeah, and so some of these analysis with Jerome is we're hoping he was going to come here to the States. He had some medical issues that he can't come anymore, but we were hoping to do this maybe potentially with snook and develop a model that we could apply to other species.
Yeah.
Cool.
Yeah, so something to talk about.
Yeah, great.
Yes,
Joe, go ahead.
What's the advantage of spawning in 300 feet of water?
Yeah, and bonefish do that too, right? It's really bizarre. So my understanding is that the pressure helps get all the eggs out and improve survival of the larvae, but I doubt the extent of my knowledge.
No,
It doesn't sound like I know very much about it, so I'll leave it there. Yeah, tough question.
That is excellent. Fantastic seminar, Luke. Let's give him a hand. We'll be around here for a bit for questions and more discussion, and thanks everybody online for joining us.
Thanks, Ron.
Cheers. Thanks a lot again. Bye bye.
I love you, Drop.
Yeah. Yeah, exactly. He's already better.